So, hi everyone, welcome to Foresight's Biotech and Health Extension Group, sponsored by 100 Plus Capital. I'm really excited to see so many of you here and lots more folks popping in through the waiting room. I think before we launch into today's meeting, two really quick announcements. One of them is just a reminder that Foresight's Vision Weekend is coming up, and it's happening for the first time in San Francisco at the Internet Archive, a rocket company, a ship, and a Berkeley Hills mansion, and in France in parallel in December at a beautiful castle. So if you're interested in joining at either one of them to meet, a conundrum of our biotechnology, of our nanotechnology, and of our intelligent cooperation group, paired with our new technology and space efforts, then this meeting may be for you. And, as someone who's a member of this group, you're definitely eligible for our subsidized ticket. So if you're interested in this, please apply, check out the program, and we would love to see you there. All right, I'm going to stop sharing now. There's one more announcement that we'll have to make, and this is Tatiana. Tatiana, if you're here, please let everyone know about your recent research efforts, and then we'll hop into our keynote. Tatiana, welcome.
Yeah. Hi, can you hear me? Yeah, okay, thank you, Ellison, for giving me the stage for a moment. Hi, everybody, I'm Tatiana. I recently graduated from my PhD from Caltech and moved up here to start a company with my co-founder here, David, as well. We're working on bringing single-cell RNA sequencing directly to consumers, and we're looking to use this very rich data set to connect people so they can share their health interventions. And so we're very new, we're at Lumen Accelerator, and we're working on developing our technology. And we're doing a study next week, so we would love to have you join, come by at Foster City and self-collect your own blood. It only takes 10 minutes. It's a very cool device. The idea is that in the future, you will do it at home. So whenever you do like a self-health experiment, it would you could look at what's happening in different cells, immune cells in your body. Yeah, so I'll just drop the link here in chat. If you guys have any questions, please feel free to contact me and hope to see you. Thank you, Ellison.
Lovely, and we'll probably have Tatiana as well at Vision Weekend. So, all right, we have lots more people joining in the waiting room. Kristen, I think you are going to have quite the full house. So thank you so much for joining. I know it's been long overdue. Thank you so much for being flexible on the timing. And yeah, we're really looking forward to your presentation. And you're gonna launch right into it. Before we have like a structured Q&A, I will. I'm assuming that we have lots of questions. So please, for our audience, if you want to ask something, make sure that you get your questions in early because we're going down the list. You're free to upvote your questions with +1. And today, Kristen Fortney will present on building a pipeline of clinical stage therapies that target the molecular causes of aging. I'll share much more information about you in the chat, and I'll be in the chat in case anyone has questions. Thank you so much for joining.
Thank you, Ellison. Great to be here. Let me just pull up my slides 1 second. Is that good?
Looks good to me.
All right, yeah, no, it's great to be here. It's a great community that you guys have built. Today, I'm just going to introduce Bioage and how we're approaching aging at our company. And it's really around building a pipeline of clinical stage therapies that target aging based on our human data sets. So Bioage is a now clinical stage company with 3 different programs that are growing, right? We have 3 programs today. I'd like us to have 10 programs in the next few years. And one distinguishing feature of the company is that we are big believers in human data. And so we've committed a lot of resource to figuring out how humans age. I think that because humans age on the scale of decades, you really need molecular data that span decades to understand human aging. So all of our discoveries and our science is built on top of our human biobank relationships. So we have partnered with some biobanks that have been collecting samples from people when they were middle-aged and onwards for the rest of their lives. So some of these samples have been stored for as long as 50 years, like blood samples in the freezer. So we have these longitudinal samples that are connected to really detailed electronic health records that track how these people aged. And our fundamental approach is really just to find what's different about those people who are already living longer and living healthier, who are already, aging more successfully than the rest of us, and seeing what's different about them on a molecular level by leveraging all the modern technologies that we can, like the proteome, the metabolome, and the transcriptome. And that's kind of the starting point for everything that we do, and a big part of our philosophy. And I'll get into that in a minute. So today I'm just going to introduce our general approach and then go into a bit more scientific detail around our most advanced phase 2 program. So I think I don't really have to explain this to this audience, but aging results in physical deterioration and death from disease, right? It's kind of still the neglected risk factor that underlies your susceptibility to all these different really debilitating diseases like Alzheimer's, like cancer, diabetes. All these things rise exponentially as you get older. And importantly too, it's not just the disease incidence, it's also the debility. So as you get older, you become weaker, your physical function declines. Your immune system becomes more vulnerable to infections like COVID and responds less well to a vaccine. And this is sort of really underlies the age state. And the promise of the science in the field, I think, as you all know, is that there can be single targets, single interventions that are directly addressing aging, and so that might help us live better and live free of multiple of these diseases at once. So they're pleiotropic in that sense. So just a few highlights on Bioage. One second. This is there. So just a few highlights on Bioage. So today we've built this target ID platform that I described to you briefly, and that's kind of like the hard piece of what we do, right? Finding out the pathways that matter for human aging is the unique thing that we do that is novel and is hard. So we have these proprietary longitudinal aging biobanks spanning 50 years of human aging, and that's where our target ID comes from. And after that, we're trying to make everything else that we do as easy as possible, because being a drug discovery company is very hard. So we're actually focused first on developing drugs for those targets that are the easiest, that are emerging from these data sets. And what I mean by that is ones that are well understood. The target biology is well understood, the pathway is well understood. And it's de-risked to the extent that there's already a drug out there that's been in humans. It's been in a phase 1 study. We know it to be safe. We know that it hits its target. And Bioage can go out and bring in that drug and go immediately to a phase 2 proof of concept trial in a severe aging disease. And that's been our approach so far. And that's how we brought in our first 3 programs. And to date, we've raised about $130 million from great investors in tech and health to advance these programs to a phase 2 POC. So just a little bit about how we think about value creation for drugs that target aging, because I think this is a problem that probably a lot of you struggle with, right? If you're really bringing in a drug that you think is going to slow aging, how do you actually approach clinical development there? How do you create value? You can't do a clinical trial that, uses thousands of people and takes decades. That's not practical. That's too expensive. It takes too long. So this is how we approach the problem at Bioage. And when we bring in a drug, we are betting on a mechanism that we think ultimately has the potential to be, to help a very large population of people for an aging indication, like frailty or like immune aging. And that means that so far we've been betting on mechanisms where, the molecule looks to be pretty safe so far. It's an oral dosing once a day. So there is long-term the potential for chronic dosing in a healthier elderly population. But that's the long term. In the near term, and this is a hard constraint, we're also only committing to those mechanisms where we can see that there's going to be an initial model indication, a first indication we can go to in the clinic that is an acute disease with a clear regulatory path with the FDA, a clear payer strategy that's valuable in its own right. And I like to say that, any successful aging drug should and could follow the development path of a statin, right? So statins today are prescribed as though they were aging drugs. If you're over 40 and you have a couple of risk factors, your doctor will give you a statin. But that's not how they were first approved. They were actually first approved for an orphan disease, familial hypercholesterolemia, and the label was widened over time. And I think this is the most practical road that you can take. And you really, usually you can, this is an important point. I think for most aging drugs, you can take this route as well, because if a mechanism is relevant to aging, it is, unsurprisingly, also relevant to many different age-related diseases. And, as you know, over the last few decades, the lifespan of the population has increased, but there's still this very large lifespan health span gap where people in the US today live until their late 70s, low 80s, but your healthy life expectancy is really into your low 60s, and then you start to accumulate more and more diseases of aging. And this is where most of healthcare costs are, and this is where quality of life is quite poor. But there are, of course, examples of healthy longevity amongst us, showing us that humans can solve this problem, right? It's living longer and living healthier longer is compatible with human biology. One of my favorite examples is Leila Denmark. She was a practicing pediatrician until the age of 103, right? So retired 40 years later than the rest of us. Brain still worked, body still worked, it can be done. Similarly, Robert Marchand there on the left set a Guinness World Record as a competitive cyclist at the age of 105. Actually, we have one of our programs, our drug programs is called BGE 105, and it's named after him. So we really believe that we can learn from humans. And the lowest hanging fruit in the aging space really is to copy what's already working in these humans who are successfully aging. And just to give you a brief history of the aging space, and probably this crowd is more knowledgeable than most about the history of this space, but this is how we think of it. It's a bit of the bioage lens here, right? And I think a lot of the people that are outside aging science don't appreciate, don't understand how new and how recent it is, right? Like aging was sort of not, even today, if you go to most universities, there's a handful of people who study aging and dozens who study any particular age-related disease. And the entire field was really quite quiet. I think a lot of biologists thought that aging was so complicated. There was not going to be any, so many different things go wrong as you get older, that there would be no single simple intervention that would have a dramatic effect. And of course, that changed in the early 1990s with Cynthia Kenyon's work, with Gary Revkin's work, showing that you could delete a single gene and have a substantial effect on lifespan. Like that you could, double worm lifespan basically with one single gene mutation, which was amazing. And I think surprised a lot of people. And then of course you see a lot of these invertebrate biologists coming into the field. I think that's still true today, where most aging biology is invertebrate biology for very practical reasons, right? Like these animals only live for a few weeks. So it's very easy to try deleting every single gene or giving them a drug and seeing how that impacts lifespan. And that becomes a lot harder as the animals become longer and longer lived, like with mice. And it's really only in the 2000s when you see a handful of things that reliably work in mice, like rapamycin and senescent cell clearance, parabiosis, et cetera. And still to this day, only a few dozen things have been tried in mice in part because these animals live so long, right? Like it's a 4 or 5 year experiment if you really want to do a full proper lifespan experiment. So it's still a short list. And at Bioage, really, I think if we want to bring therapies to humans, we have to understand how humans age. And that's a hard problem because we take even longer to age. We take decades to age. But one of the reasons it's so important is because the way we age is so very different. Like a lot of, invertebrates, they die due to causes of death, insofar as we know. In some cases, they're still mysterious, but they're linked to things like intestinal failure, which are not bottlenecks for our survival. And even inbred mice, right? They die almost exclusively of cancer. An old mouse will never get Alzheimer's. An old mouse will never get heart disease. And that's why these models are so poor when we try to translate things that work in these animals to us. And humans, of course, have this very different spectrum of causes of death. However, we think that with today we're in a very unique position to be able to understand how humans age. There are these biobanks that have these, really precious samples collected over decades, and that's, I think, the right data set to understand how humans age. And with the modern toolkit of omics technologies, you can really go and enumerate all the molecules in those samples and really understand how those people are aging. Like just to give you an example, like the proteomics that we use today, you can see, 7,000 proteins in a blood sample. And that's new, right? Like a decade ago, you couldn't get nearly that many proteins when you're looking at a sample. So the convergence of, I think, these technologies and the right algorithms to analyze the data from these technologies, coupled with these sort of decades of human aging data, give us a view into what's most important for human aging. And then, just briefly, I've already said this, but this is showing you the distribution of age at death in one of our cohorts. And as you can see, there's a huge spread, right? Like some people only make it to their late 50s, others make it as late as 110. And really, we're looking for the molecular factors that explain the differences. And that's the starting point for everything that we do. So just at a very high level, this is the platform that we've built. It's sort of like a human first, data first discovery platform. And we're starting with, again, these natural human aging cohorts, these natural human experiments with 50 plus years of follow-up. We're characterizing these samples. We're using every modern technology we can. And so far, that's the proteome, the metabolome, and the transcriptome. We're using modern algorithms to figure out the most important pathways and networks in these data sets. And then another important point is that we do validate everything that we find here in old mice. and particularly in those sort of, like, so as a company, I think this is an important point. We focus on a few different therapeutic areas. A couple of those are muscle aging and immune aging. And we focus on those because we think that, because those happen to old mice also. So like, I think that there's a lot of, exciting signal in our human data for brain aging too, but I have no idea how to validate that because the mouse models of Alzheimer's are so terrible. So we're big believers in also looking at those phenotypes that also naturally happen in an aged animal context. So if you have a really old mouse, just like us, its muscles are going to be, function not so well. It's not going to run as well in its wheel. Just like us, its immune system is going to be very vulnerable. It will be much more likely to die if you give it COVID or influenza. And that's one reason why we like those models. So we have thousands of old animals that we validate our pathways in. And then once we believe that, a particular asset is affecting aging in humans and in this translational animal model, then we in-license a drug. So we are, to date and for the next few years, really, really buyers and looking for drugs that are clinical stage or near IND, where we can go as rapidly as possible to a human proof of concept. And just a snapshot of where we are today in terms of the data and the algorithms that we're looking at. So today we have about 1.5 million clinical data points from our cohorts, 65 million molecular data points, 50 million molecular relationships from all the data sets that we put together. And the bandwidth, in terms of throughput, we're looking at about 20 different pathways per year, company-wide. And to date, we have 3 clinical stage assets, 1 discovery stage asset. And usually we've got a rotating set of 10 assets under diligence that we're looking at to see if they might be important. And we like to think of this as really like de-risking at every step, right? If you want, your molecule to work in humans, you better believe that it's relevant in humans. You have to start with human data. And 2, revealing underlying pathways. When you start with data sets, it's really hypothesis free. You're letting the data tell you what the most important pathways are for human aging. 3, targeting key disease drivers. There's a lot of, network algorithms and, genetic data to bring to bear to figure out the most important targets, the most important pathways in these data sets. 4, we use these naturally aged mice for all of our validation initiatives. And then 5, very importantly, if you bring in a drug that's already been, in humans, that's a huge de-risking step because you already know that it's safe. You already know that it can hit its target in human context in the way that you need to actually impact the disease. So just to give you, a snapshot of one of our programs and how it progresses through the platform. So the drug that we brought in most recently is a receptor of the Aprilin agonist. It was a drug that Amgen was developing, and it's already been in phase 1 studies of about 200 different people. And this is just, a snapshot of some of the platform data that made us really excited about Aprilin signaling in the Aprilin pathway. So on the leftmost panel here, you can see some of the variables that we looked at in our cohorts, right? And I didn't mention this earlier, but what I think is really important about the cohorts is that they have information not just on how long people live, but how healthy they are when they're older. So we have, for the participants in our biobanks, their grip strength through time, their walking speed through time, their cognitive function through time. So when we're looking at Aprilin, some of the variables that were really important here were survival, but also physical function ones. So you can see in the second panel that if you define a muscle aging score that incorporates information about longevity, but also muscle function, then Aprilin, the protein itself, comes out as one of the most significant signals there. And you can see the actual effect size on the bottom there. So if if you look here, you're looking at your Aprilin protein levels compared to other people your age. This is for middle-aged people. And as you can see, there's a pretty linear relationship where the more Aprilin you have, the more likely you are to live long, the more likely you are to have good physical function when you're older. And in the third panel here, we're looking at the network context of Aprilin. We think it's important to not just look at the protein itself, but also the set of proteins it interacts with and performs its function with. And as you can see here, if we look at this network component, there's a very strong pathway signal for mortality. So that sort of gives us extra confidence here. Now, when we got excited about our data, we reached out to Amgen and they gave us some of their drug. We put it into various old animal models of muscle aging. A lot of interest, it's worked in everything we tried. But the one piece of data that we think is the most important for the clinic that got us the most excited about the clinical setting is this picture right here. And what this is it's a muscle atrophy model. So you take an old animal, you cast one of its limbs, and you wait 3 weeks. And you get, just as in older humans, you get substantial muscle atrophy. Really, in these animals, the muscles sort of decrease their weight by a half. And as you can see in the figure here, that's almost completely protected by the drug. And that's, pretty exciting to us. There's a lot of important acute clinical indications where older people are hospitalized and experience substantial muscle atrophy. So based on these data, we in-licensed the drug from Amgen. So we have 3 different platform-derived therapies now, BGE-175 for immune aging, BGE-117 for aging anemias and muscle aging, and BGE-105, the one I just introduced for muscle atrophy and muscle aging. And I'm going to spend the rest of the time just talking in detail about our immune aging drug, because this is really our most advanced program where we're in the middle of the phase 2 trial currently. So this is a drug that we in-licensed from a company called Shionogi. It resets certain aspects of the older immune system. And in multiple preclinical models, it really protects older animals from pretty much, every viral or bacterial challenge we've tried. And that includes COVID-19. So here we're going first to an older COVID-19 population. As you know, this is a disease very clearly driven by immune aging. If everybody had the immune system of a 20-year-old, it would be a very different pandemic. And we're going to get our proof of concept here. So just a snapshot of this drug. It's an inhibitor, a very potent inhibitor of prostaglandin PGD2 DP1 receptor signaling. And here it's an oral drug, so it's just a pill that you take once a day. And it actually corrects 2 different aspects of immune aging. So as you get older, your dendritic cells don't migrate as well as they should to wake up your T cells in response to a viral or bacterial challenge. And also you get way too many infiltrating neutrophils into older lung in a way that's damaging. And this mechanism, if you block this receptor, it actually corrects both of these back to sort of younger levels. And development to date, so this drug has already been in over 2,000 patients in the original indication. So the company Shionogi, where we got the drug from, was originally bringing it forward for seasonal allergies. So it's a very, very safe drug. It's already been in, over 2,000 people in phase 3 studies. They stopped development because it was no more like effective than a generic antihistamine. There wouldn't have been a market for allergies. But our data showed that it would be relevant to these very different indications around immune aging. So here, just to give you an example of our strategy of starting with something very acute and very focused and how that expands over time, we're going first to older hospitalized COVID-19. The next indication on the map would be, viral pneumonia independent of the cause in a hospitalized setting, which is a very severe condition in older people. And then ultimately, we think that this molecule could be helpful earlier in the course of disease. So it could be a drug like a Tamiflu, like an antiviral, and there are very large established markets for influenza RTIs, where there's still a lot of unmet need, because even though most people receive antivirals early in the course of disease, they just don't work very well. So I already briefly touched on this, but importantly, this is affecting 2 different, it's a dual acting mechanism that's really correcting 2 different aspects of aging-related dysregulation. So it's directly improving your dendritic cell migration while also suppressing your overactive neutrophils in an older population. And I'm going to show you some data now from one of our collaborators, Stanley Perlman. So when we started working on this molecule, we did some work with influenza challenge, but then the pandemic hit and we thought it might also really be helpful for COVID. So we reached out to Stanley, who's a COVID expert. He's developed a lot of the preclinical models that other labs use. He sits on the FDA advisory committee that approved the Pfizer vaccine. And he stuck it into a bunch of his models. And there's just a quote there from him on the bottom right. But we have a paper with Stanley submitted now under revision. And the real takeaway here is that it works very robustly across a range of different respiratory pathogens. Like, it's not a direct acting antiviral, it's sort of improving the older immune system. It's a host directed mechanism. So you might expect it to have a very broad response, and it does. So this includes 4 different models of COVID-19, for SARS from a decade ago, for influenza, for RSV, there's a very strong response in all of these models. And just to show you the data around COVID for the, because that's the current clinical trial that we have right now. So this is data from Stanley's lab. And what we're doing here is at days, you're taking some middle-aged mice. And at day 0, you're giving them a lot of virus. And then at day 2, when they're already quite sick, they start to receive our drug. So it's kind of the, the mouse equivalent of a hospitalized setting. And as you can see in these older animals, without drug, that's the yellow line, they all die. And there's close to total rescue on the drug. And this is really very striking. And at the same time, you see, if you look in their lungs at day 5, there is 10-fold less virus in the lungs of these mice, even though this is not, a direct acting antiviral. And this is looking at another COVID-19 model. We looked at both the mouse adapted virus as well as the mice that were humanized with the human ACE2 receptor. And you see, basically an identical curve, right? No matter which model you're using, the mice without the drug, they all die, and there's almost a total rescue on the drug. And interestingly, you see the same thing if you look at, for example, SARS, the coronavirus. So it's like totally different, virus from a decade ago. Very identical response, right? So this kind of drug and this kind of response doesn't really care about the precise sequence, doesn't even really care about the virus itself. It's really sort of correcting the host immune system. So right now we are in the middle of our phase 2 trial for COVID-19. This study is going on in the United States, Brazil, and Argentina. And we're basically taking older patients who are hospitalized. They're not yet on high flow oxygen or intubations. They're severe enough to go to the hospital, but not yet, on a ventilator. And it's 14 days of dosing, after which we're looking at the proportion of subjects who've died or progressed to respiratory failure. So importantly, in this older hospitalized patient groups, still upwards of 25% progress to poor outcomes, like after this stage. So we're looking to see if we can get that number down with this drug. At the same time, of course, we'll also be looking at secondary endpoints like safety, the COVID ordinal scale, viral clearance, and also biomarkers of immune aging from our platform. And just as a reminder, like the incidence curves we've seen with COVID, we see every year with the flu, right? Like these, as you become older, you're really more vulnerable to pretty much any viral and bacterial infection. And we think this mechanism has the potential to be helpful in all of these contexts. So that's just a brief intro to one of our programs. And with that, I'll just end to say that our mission is to develop a pipeline of treatments that extend healthy lifespan by targeting the molecular causes of aging. And we've got a great growing team in aging biology, AI, and drug discovery, and we are hiring for a number of roles all the time. So a few of them are listed at the bottom here. And if you want to see the full list, you can go to jobs.lever.co/bioage. Many of these too are also remote optional. And there's a couple of pictures of the team from a recent sailing trip. Yeah, thanks.
Fantastic. Thank you so much. Wow, that was quite the walk. Thank you. If you don't mind, I'll stop sharing your screen. You have a variety of questions already collected in the chat. I hope you don't mind if we jump right into it.
Yeah, go for it.
Okay, good. Let us know if you need a little break. That was a lot of information. Thanks a lot for sharing. We have the first one, Nicola Markov.
I hope my microphone works. Yeah, I wanted to ask something a little bit from a philosophical standpoint. I don't have a strong opinion about it, but looking at exceptional longevity, we're definitely looking at the edge of the curve. So we're looking at people who have very specific circumstances in multiple omics, in multiple combinations of, everyone has their own situation that has allowed them to have the exceptional longevity. And if we're trying to find a drug that will be acting in the middle of the curve and be able to help most of people, how do you think this to work? Is looking at the end of the curve going to help us to find the drug that works at the middle of the curve, or is it another approach going to be more appropriate, or should we be doing something that personalized medicine?
Yeah, I think there's a lot of ways to generate interesting hypotheses about which drugs and which programs you might want to pursue. one reason why we like to look at these sort of dynamic omics, like the proteome, for example, right, is because in a way they're integrating your genome, your environment, your diet, your lifestyle, right? So for example, we discovered, and I showed you guys that people with higher apelin levels were living longer, were living healthier. And that protein might, they had elevated apelin, but that might have been because they were born that way with the right genes, or maybe they just exercise a lot, right? So that sort of signal, I think, is a good starting point, right? I do want to emphasize that it is just a starting point. After that, you still want to go and reassure yourself that it's not just a biomarker. You want to actually intervene in a model and see that you can improve the phenotype. And that's why you have to do the in vivo experiments as well. Yeah.
Looks happy. Okay, great. Creon.
Oh, hi, thanks. Wonderful presentation. I have a simple question and a complicated question. The simple question is the COVID-19 intervention in the hospitals that you were describing, getting started on, is that going to be double blind?
Yes, it is. Yeah, double blind, placebo controlled, all that. Yeah.
Super. Okay, then the other question is more of a general one, which it's the same question I seem to have in my mind whenever I hear these kinds of software-based, omics-based anti-aging companies present. And you're obviously, one of the leaders at your company, but still, a lot of people kind of in this space, right? It's like throw a bunch of data and a bunch of machine learning and come up with a target molecule and then see if we can, raise it or lower it or modify it or modulate it. Okay, how do you figure this out? Like there's 50 million relationships, you said, between these molecules that you're tracking. And you found like this one where you intervene and it seems to modulate immune aging to maybe set back the clock a little bit. But presumably there's something upstream of that, which is modulating that. And there's like correlation does not equal causation. And also, even if you have causation, it doesn't mean you have root causation. And I'm wondering, are we going to just need 100 different interventions for every little thing we find that is part of aging? Because we haven't really unraveled the causality and the basic principles since we're using black box data analysis, or are we going to somehow either be able to use the black box or our own knowledge to unravel the root causes and try to modulate those instead of every little molecule that looks promising?
I mean, my intuition there that, drug discovery for aging is not really going to be fundamentally different from drug discovery for heart disease over the last couple decades, or drug discovery for any other area where pharma is really well established, and where, yes, it is going to be a bunch of mechanisms that might in some cases be additive. And there's no sort of single root cause master switch, at least that we've discovered to go after there. I mean, my perspective, I guess, on the machine learning and on the omics is that this is and I'm bullish here, of course, for obvious reasons, but I think this is the way that we do modern science, right? I think that these have been new technologies for the last decade, and they are going to be under the hood at pretty much every future, biotech company, because when you're analyzing a complex biological system, of course, you want to measure everything. And of course, you need these algorithms to bring to bear just to see what's going on. And I think you want to really ultimately view it as hypothesis generation, right? It's another way of generating ideas about things you might want to switch and try and see if they work. So it's not, it's not the whole thing, right? But it is where your hypotheses come from. And in our case, go ahead.
I hope you come to the Vision Weekend, or that at some point we can sit down and talk for 45 minutes about this, because it's a big topic, but thank you.
Sure.
All right, next one up, we have Ronald.
Hi, Kristen. Brilliant talk as usual. Thanks for giving it. I have one technical question, one sort of comment. So the technical question is, are these naturally aged mice behind the pathogen-free barrier?
Yes,
are they exposed to pathogens?
They are not exposed to pathogens. Yeah.
Oh, okay.
Yeah.
And then the kind of comment is, so you have an intervention going on with the immune system, and people like Kirkland and Niederhofer and all have senolytics, which are probably downstream of that. Any options for combination, somewhat along what Creon was asking?
Yeah, no, I think that's really interesting too, right? Like the aging immune system is at the center of so many different things that go wrong as you get older. And there's so many different changes. So I can tell you from our data sets that one of the strongest signals that we saw, like there was a huge signal for neutrophils, and that was sort of partly behind why we went with this particular mechanism as our first mechanism. But I expect there's going to be a lot of other cell types that matter. And yeah, with the senolytics in particular, we haven't tried combination therapies, but I think that would be interesting. Certainly with these models, it wouldn't be enough because you already see a full rescue on our drugs. So we'd probably need a different model to explore that. But I do believe that combination therapies are ultimately where these are going to go. Yeah.
All right, lovely. Next one up, we have Nir.
Yes, well, I have a comment, but I should say first, Kristen is not only a great colleague, but she became really a great leader. And you should know that she's doing a lot of things for this community, much beyond caring for her company. And Kristen, that's just terrific. And we're just blessed to have you. And that was as good talk as ever, A+. I made just one comment that, you know, we are at danger of setting ourselves to an impossible task with the FDA. I don't think that sarcopenia is really a good target for drug development. I think it's too late. Just as much as frailty, in some way, it's too late. The whole idea that we have is to prevent the aging, not to treat the aging. So we have to think of how, and you know, I said, you know, imagine, Kristen, imagine your drug is testing sarcopenia and it's negative. Well, you know, it still can be a great drug to prevent it. And what will the FDA say? You didn't prove the principle. So we have to take care of those things also.
Yeah, I think you're right, Nir, right? Like we at Bioage are really starting with, you know, the easiest things, right? So where we think it's an aging mechanism, but also we think it's going to treat an acute disease. And there probably are going to be some very important mechanisms that do not treat acute diseases. And what do you do then, right? So I think we also need innovation on that side too. Yeah.
Okay, great. I have another question, which I am going to read out because I'm not sure if the mic is great from the participant, but what protein system are you using and others you know of?
Proteomics system?
Yes, Johnny, do you want to provide some context?
Yeah.
I don't know if your microphone is off.
Oh, there, it's on now. Yes, thank you, Ellison. That was my question.
Yeah, so we've been working with Somalogic so far. We're also trying out some of the newer technologies like SEER. So yeah.
Oh, and did you say SERA? S-E-R-A?
SEER, it's S-E-E-R. Yeah.
Okay,
Yeah.
I'll find them. Thank you.
Great. We have Tinka next and then Robert. Okay, while she's unmuting, Robert, you can just go with your FDA comment if you want. So it was just.
I'm now unmuted. Okay, so sorry. I have a question regarding BGN and Aprilin agonist drug. It is known that that Aprilin expression is high in many kinds of cancer, and some people believe that it can lead to cancer progression because of increase of angiogenesis and proliferation. And is it possible that this molecule BGA could that is developed to treat muscle atrophy could lead to increased incidence of cancer? Did you check that?
So we haven't done like lifetime carcinogenicity studies in mice, if that's the question. But I would point out that from the discovery perspective, like we're looking at people who have higher Aprilin levels from middle-aged and for their entire lives. And there is no observed higher cancer incidence in this human population. I think there are a lot of things that can promote cancer once you have it. A hypoxic environment is another example that don't necessarily predispose you to cancer if you have them beforehand. Yeah.
Okay, thank you.
Lovely. Okay, Robert, you're next. Okay.
Thanks. Yeah, it was just meant for the chat, the question that I put down.
Okay, got it. So Kristen, you now also have to multitask and monitor the chat because there's an entire second conversation happening. No, don't worry about it. I'll send you the chat later in case you want to follow up. But we have one for you, which is from Juanita.
Hi, Kristen. That was a great talk. My question is specifically about the biobanking and what kind of samples you're actually analyzing, and also how are you getting 50 years of samples if you're a relatively new company? How do you get access to those samples? Basically, are you looking at microbiome data? Are you just getting tissue samples? Are you getting blood samples? You know, the whole gamut.
Yeah, for our cohorts with the longest follow-up, it's basically just blood. So it's a great window into the immune system. And as you know, from parabiosis, blood goes everywhere and can benefit multiple tissues, but it's not like, you know, because these are samples that they were collecting from healthy people. So there's not going to be banked brain samples. I wish there were banked muscle samples. We have some cohorts where there's like less follow-up where they have, you know, muscle and frail people, but it's basically blood. It's blood through time, blood cells and, you know, plasma. And oh, and your other question, yes, we haven't been around for 50 years. there were a few, there's just a handful. Like I think biobanking is very popular today, right? Like everyone is biobanking. That was not true 50 years ago. That was especially not true for healthy cohorts, you know, like people collecting cancer samples or whatever, much more common. But there were a small number that did that. And so we've very intentionally gone out to find every biobank that has the right samples, that has the right clinical follow-up, and have built relationships and partnerships. Yeah.
Have you looked at all at microbiome samples from people over time?
Yeah, we don't have that. It's just blood samples that's available for these people. Yeah.
All right, thank you. Okay, so again, a question for, sorry, a reminder for the chat. Stop me talking if you have other questions. You can do so by asking questions. And until then, you know, I'm going to ask a few standard questions that we usually people really find valuable for having you answered, which is, you know, the first one will be more about what do you see like the more long-term trajectories of this work being able to accomplish, and then, you know, what can people in this group do to help? But let's tackle the first one first. You know, what do you see on the horizon for people? You know, what are interesting bottlenecks, opportunities where people could, you know, where new work still needs to be produced, and then the next one will be more actionable.
Yeah, so my feeling, I guess, you know, is that so far as longevity hypotheses in the clinic, very few of them have been tried so far, right? Like it's a new area. There's only a few companies going to clinical development today. And honestly, what I think that means is that there's going to be a lot of low-hanging fruit, right? Like if we've, you know, I said earlier that, you know, most of the genes and drugs that make worms live longer probably won't work for us. But I think the takeaway from all that work is that a lot of things worked. I think there were, you know, several hundred different things you can do to a worm and reproducibly make it live longer. And I don't think we're optimized in that sense either. So I bet there are going to be a lot of different things you can do that will impact lifespan and health span. And so in terms of, you know, opportunities, I think that, you know, like I'm a big believer, I guess, in sort of brute forcing. Like I think there's a lot of easy things you can do to sort of start to scoop up some of that low-hanging fruit. And the lowest low-hanging fruit, I think, is learning from humans that are already doing it. So that's our biobank approach. That's also, you know, Nir's work on centenarians, which I think needs to be done at the right scale. So there's a few, I'm excited about those kinds of projects, or even just, you know, longevity testing of interventions in mice, like at scale, you know, like the NIA ITP is a wonderful program, but I'd like to see that multiplied like 100-fold, right? Like the NCI, for example, you know, has put tens of thousands of compounds into mice to see if they cure mouse cancer. So I'd like to see an agent initiative at the same scale.
Oh, wow. And what would someone, you know, who newly enters the field, like apart from just, you know, applying to work at Bioage, what are other bits, you know, that they could take on there? Because that seems like a really hard one, the program to set up, you know, it's like, it's quite the holy grail.
Well, part of it really is just of seeing what they've done for other disease biologies and applying that to aging. So I still think we are in this position where aging is underfunded and there aren't enough people working on it. And even sort of the more sort of simple transference of structures and knowledge and, you know, right, like from other areas. It's like, so I think that shouldn't be like, I guess, too hard to get into in the new area. But you also asked how people can help Bioage, and we are happy to hire people involved in the community as well. So I already put in a plug for that earlier, but just to mention it again. Yeah.
Yeah, again, you know, like I guess just to come back to that question a little bit. Like, I guess the group, we're like gradually onboarding more and more people. We have like an application board, and we're gradually drawing more people in, but they often are just like, okay, what is the best way in which I can really be of use? And I think like one thing that we really try to do is also figure out how can the community help like really promising projects a little bit more. So like apart from just hiring, what are other bits that you're looking at with Bioage, and what are potential ways in which the community could help on a large scale? I mean, there's obviously there's folks that may want to apply to work there, but there's also investors. There's also folks that work more on the policy angle, and there's more folks that do more self-experimentation. There's a variety of like a broader, I guess, selection of people, and like it seems like many folks could help in different ways.
Yeah, no, that makes a lot of sense. I mean, I think the policy angle is important too. As Nir mentioned earlier, it's like, what do you do with you have an aging drug that doesn't fit into sort of the conventional narrative around treating an acute disease, and how do you measure? And honestly, I think that that's going to involve a lot of different stakeholders and companies working together to come up with good surrogate endpoints. I mean, like, for example, right, even frailty today, frailty, or you don't see a lot of companies doing drug development there. And one of the reasons why is because, well, first of all, the FDA doesn't even like it. But even if you were going to do it in Europe with the EMA, where I think they're more open-minded about that, that the endpoint that you'd be trying to move is a 6-minute walk test. And this is an incredibly noisy, heterogeneous endpoint. And, you could probably do a lot better with wearables data, right? Like, I think surrogate endpoints in general, right, can help you run more faster, smaller, more efficient trials. And that will lower the barrier to making a lot more companies experiment. Because right now, if you really want to do like a well-powered frailty trial, it's so expensive and it takes forever, right? So there's not a lot of people trying. So I think, the selecting and adopting the right surrogate endpoints and convincing regulatory bodies that, this is useful, will also open up innovation in this space.
Yeah, much definitely heard on the surrogate endpoints. I have had a follow-up, but let's take Bolek first, who just posted a question to the chat.
Yeah, hi, hope you can hear me. Basically, I have a quick question regarding more of the investor community and your relationship with them, or like more, if you're pitching to them and you're one of the high-profile companies, basically, do you feel like the pitch longevity thesis is resonating more over the time, or do you feel some like shifts there, between the tech VCs like Andreessen Horowitz and then some biotech VCs? What do you feel is the how does that work, basically? Yeah.
No, I think it's been getting a lot better, right? I started my PhD in 2007. It was like way unpopular then. And Bioage, it's an older startup, right? So we raised our seed in late 2015, and that it was post Calico, right? But there wasn't a whole lot else going on, right? So and especially in the last year, I would say there's been a lot more traction. A lot of investors, even conventional biotech investors, are interested in this space. Large pharma is more deliberately interested in this space. So it feels like we're at the beginning of an important movement here. Yeah.
Thanks.
And in terms of like building out the movement a little bit, because I think that the first time I heard about your work was also a few years ago. I think you received, was it a Buck Prize for your work, right?
Or Glenn, maybe. Yeah.
Like so quite a long while back, and since then, like, your work in Bioage has really like totally exploded. And so I'd be super curious to just hear a little bit like your assessment of like what changed in the past few years, and like, are there any things that as a community like would be good to have in our minds as we grow the ecosystem forward? Because just from like a local view of how applications to this group have exploded, I think that the interest that has really picked up during COVID, and then with I think also crypto really moving into the space, and with software getting much better, like there was a conglomeration of factors that I think has really led to an explosion in the space. And I think just like having a little bit of your local view of like how that happened, and like what would be like, I guess, trigger point to make the community grow in like a healthy way would be really useful.
Yeah, for sure. No, hard question. Yeah, COVID, I think, is a good one, right? Like, because of COVID, well, there was a biotech boom in general, right? Because science saved everybody, vaccines saved everybody. and at the same time, I think it really brought immune aging to the forefront. I think people realized that it happened, but didn't quite understand the impact until you see what the huge effect size that there is for COVID deaths, right? So I think that you no longer have to make the case that immune aging is a bad thing, that it has these bad consequences, right? So I think that side of it, I would say. So both the more money in biotech in general, but and for more sort of longer term bets in biotech, like longevity science, things that are newer, like newer modalities, but also new indication areas, right? Like aging. And yeah, no, I think that the crypto boom has been really interesting too. There's certainly a lot of people that are doing philanthropic projects right now. So it's very good news for the space, which I think is still underfunded, right? But better now.
Yeah, slightly better. And then, I think one bit that at Foresight, at least, folks are quite excited about are like the intersection of different scientific areas and technologies. And so, for me, for example, what's interesting about this group is that, I can really see how there's like a really big community formed, like there's a really big ecosystem now. People are really like more in the startup world with their project, much more so than even like, let's say, 6 years ago when we did like a few Foresight longevity technical workshops, and it was, it felt much harder back then. But I think that another group that we run is the molecular machines group, so that is more on the nanotechnology angle. And so there it is really still very much basic science. And it's sometimes really hard to square that kind of like that gap between, I think many of those projects already could be almost on a startup level. And ideally, the molecular machines group, like the nanotech group that we have, is in 5 years, where the biotech group that we currently are in is now. So ideally that, there's a lot of projects already that, even though they still look like basic science, could maybe already be startups. But it's really hard to bridge that divide between folks in really basic science and academia, because that's not often where VCs are looking, but those people don't often know that their stuff could already be startup ready. So, I wonder a little bit, like, how do you see the intersections, I guess, between more with a little bit more researchy work still, and like, are there different ways in which one can help them, become a scientific project faster? Like, you as someone who's clearly done the divide in a fantastic way and sped things up massively, like, do you have any tips for how to go about this?
Yeah, no, you're making a really interesting point, right? Because that's been another trend in the last few years, where there's, I would say, more in biotech at least, right? Like more sort of founder-led companies where people are coming, straight from academia or more from a non-traditional background like tech, right? And there has been, increased capital willing to bet on those people. And that class, we're seeing some of the first successes just in the last couple of years, right? Like with Ginkgo's IPO, SPAC, right? And Recursion's IPO this year. So I think that's a growing segment as well. And you're right. Like, I think a lot of people in academia don't realize that you can still do like science in the context of a small startup, right? And that can be the right environment too, for risky science. Like when I raised seed for Bioage, a long time ago now, back in 2015, it was, a couple $1 million, right? That was sort of the first amount of money. And I talked to other academics and I was like, well, and it was basically for a science project at that point, right? It was to go out and like close all these partnerships and generate these omics data sets and like show that there was signal there, right? And that was still kind of an unknown. It was like, there was a lot of reason to believe it would find interesting targets, but it was still a big unknown. And I think it's still true that if you're an academic and you're writing a grant for your risky science project, like, good luck. I mean, you have to convince a committee of people at the NIH, and they all have to think that it's not terrible. And with sort of early stage stuff, I think people are willing to take on additional risks so long as there's the matching potential for a massive benefit, right? So I do think that some of these even sciencey things can be sort of moved into the startup world. And there's, I would say, growing interest in sort of supporting those kinds of entrepreneurs, but still a gap. Yeah.
Yeah. And then, it's also about, are we, should those scientists then also become the founders of those companies? There could also be an advising role, right? Like, there's something still also to be said of maybe pairing them with an entrepreneur in residence and then with another funder. So I guess, there was some appetite in our community back in the days to just do almost like a little bit more of a matchmaking function there, but maybe let's see. It's definitely a hard space to bridge. One question that I still had is, you clearly, also work a lot across sectionality with other fields, in computer science, for example. And so, if, let's say, for example, someone from a different group was watching this, like we have a computing group as well, and then we have the molecular machines group. So are there any areas basically outside of the immediate biotech area where you're just like, hey, I really wish we had this tool. We don't, but someone, like working in an entirely different scientific discipline should really make this, because this would really speed up our research. Like, are there any, let's say, more cross-disciplinary, cross-silo requests for other research areas where you're just like, hey, look, we really need this to get going?
Well, tools are really important, right? I mean, as I mentioned earlier, like the proteomics, how that's changed in the past decade, I think that's been like really key to understanding what's going on in these biobanks, and we're still not there yet, right? Like I think that's still in some sense, there's a lot of companies right now with that might get there. So other, I would say, foundational technologies and also algorithms to analyze those are, I would say, also very important. Yeah.
Okay,
tools.
Well, those are already plowing ahead. Okay, unless anyone has any other questions, I think those are, like the general foresight related bits. I really just want to thank you for taking the time for joining today. It was a really fantastic presentation. I think you covered a lot. I would love to share your slides with folks in case you're interested in this. And I think you shared, or maybe a final question of just like where should people go to if they want to find out more? I know that you shared on your last slide just a little bit more of your contact information, but what's maybe like an easy way to reach out to you?
Yeah, sure. My email is kristen@bioagelabs.com. We have pretty regular updates about the company now on social media too, on like Twitter and LinkedIn, and our jobs and news are always going to be linked to from our main website as well at bioagelabs.com. Yeah.
Okay, lovely, fantastic. Well, then just a reminder from my end that if you're interested in joining Foresight's Vision Weekend on December 4 and 5 in San Francisco and December 11 and 12 in a castle in France, with lots of people in this group here and lots of people across our groups, then please feel free to look it up on our website and apply for a subsidized ticket. There's definitely some available. And yeah, I hope to see you all there, and I hope to see you in our next virtual presentation. So thanks everyone for joining, and Kristen, thanks a lot for your time. Really glad we finally made it.
Thank you, Ellison. It was fun. Yeah.
All right. Bye, everyone.